Vikrant, and on demand of some of my degree students from BHMS, BAMS, PharmaD and BPharmacy, I am making a video, this video lecture that will continue some points, okay, a very important point from a part that is called as immunology. Okay, so immunology is a subject for the degree students, so for them I'm just making introductory part, and in detail we will study one antibody antigen detection test which is called as the ELISA test. In PUC second year or in lower standards, we have studied that HIV or AIDS is a disease. AIDS is a disease which is caused by HIV virus, and this virus is detected by a test which is called as ELISA. So here on the demand of those students, I am preparing one short video where I will explain ELISA, and along the ELISA test what are the different types of ELISA, so we will study that in detail. Okay, so let me start with basically what is immunology. Immunology deals with, immunology is a subject which is going to deal with the immune system of the body. The immune system of a body is a very complicated thing that we will study in detail as the points or the questions are demanded by the students. So immunology is basically the defense mechanism or the immune system of our body. Here basically in the next topics we will also study what is or what are the different parts of immune system. What are the different cells? There are many cells that are involved in the immune system. There are WBCs which are involved in the defense mechanism. In WBCs also there are neutrophils, eosinophils, basophils, macrophages, dendritic cells. Okay. Then there is also the types of immunity. What is innate immunity? The immunity that is present since birth. What is acquired immunity? The immunity that is derived after the birth. What are the different parts of innate immunity? What are the different cells that are involved in the acquired immunity? How the antibodies are produced, or what are the antibodies? Okay, antibodies are basically the proteinaceous substances or the proteins which play an important role in basically counter attacking the antigens. What are antigens? Antigens are all those foreign substances which when enter inside the body, these antigens might cause some infections which are the pathogenic antigens, or there might be some antigens which do not cause or harm the body. So all antigens are not harmful, that is all antigens are not pathogens, but all pathogens are antigens. Let's start with the first test, okay, or the first introductory part, that is ELISA. Now before starting this, you should know that antibody is proteinaceous nature. There are 5 types of antibodies that the human body produces. There is and the antibodies are also called as immunoglobulins. Okay, globulins because they are proteins, immuno because they are the part of the immune system. So there is immunoglobulin A, immunoglobulin D. Okay, A is also called as alpha, D is also called as delta, immunoglobulin M which is called as mu, then immunoglobulin G which is called as gamma, and immunoglobulin E which is called as epsilon. Okay, so there are 5 types of immunoglobulins. These antibodies basically they have a site in them by which they are going to go and bind to the antigen. Okay, so that antigen binding site in the immunological language, it is called as a paratope. What is called as? It is called as paratope. And antigens on their surface, okay, they might contain proteins, oligosaccharides, that is sugars or glycoproteins, and especially so those sites, okay, on the antigens they are called as the epitopes. So epitope is a site on antigen that binds to the paratope that is present on the antibody. So when the antigen antibody bind to one another, a complex is formed that is called as antigen antibody complex. Okay, this antigen antibody complex is only formed when the antigen and antibody sites are complementary to one another, and this is the basic thing which is exploited for the detection of whether the antigens are present in the body or antibodies are present inside the body, and that test is called as the ELISA, which when we expand ELISA, it is enzyme linked immunosorbent assay. Okay, now why the name enzyme when I have not discussed anything about the enzyme? We only know that there is antigen antibody reaction, but here it is an enzyme linked immunosorbent assay. What is that? We will study. There are the different types of ELISA. Now ELISA is basically used for the detection of, listen the words properly, qualitative detection of proteins or quantitative detection of proteins. Okay, then ELISA might be used for the detection of both. It can be used for the detection of antigens, or it might be used for the detection of antibodies in the human sample. The human sample can be blood, or it can be the serum samples of the humans. Let's see what are the different types of ELISA. ELISA is indirect ELISA. ELISA can be sandwich ELISA, or it is a competitive ELISA, and there is another variation or a modification of ELISA which is called as Chemiluminescent Test. Okay, now why it is called as enzyme linked? We know that there is an antibody and there is an antigen. When one antibody is conjugated with an enzyme, okay, what is happening over here? The antibody is conjugated, that it is bind with an enzyme, and it is a conjugated enzyme that is present over here. And for this enzyme, its specific substrate is added. Okay, this substrate, when it reacts with its enzyme, okay, the enzyme substrate complex is going to give a color change. A specific color is going to form, and that is going to indicate whether the antigen antibody complex has formed or not. Okay, so this substrate, as it gives color, it is called as a chromogenic substrate. Okay, so it is a chromogenic substrate. Now let us study how the indirect ELISA is going to work. Indirect ELISA is basically going to detect whether the patient sample contains antibodies against the antigen. Suppose if a person is infected by an HIV virus, okay, now naturally against these viruses the person's body is going to react, it is going to defend, and in this defense mechanism the person's B cells, beta cells, or the plasma cells, they are going to produce proteins which are the antibodies, which are going to act against the antigens. Okay, so huge amount of antibodies are not produced, less amount of antibodies are produced, and these antibodies, whether they are present in the patient's serum or not, that we are going to check. If the person's sample, blood sample, contains the antibodies, it means that the person is infected with the HIV. That is, that antigen is present inside the body of that individual, and that's why the person's body is producing the antibodies. That is going to indicate. Okay, so here let's study indirect ELISA. Now suppose this is a microtiter well. Okay, we all know, we are from microbiology background. What are the microtiter wells? Okay, in the microtiter wells, antigens are coated. What are present in the microtiter well? Antigens are coated, that is, antigens are present. To these antigens, okay, what we are going to do is that we are going to add the sample of the patient which is containing the antibodies. We will now label these antibodies as AB1. So when there is antigen and when the antibody comes and it is going to bind with that specific antigen. Okay, now to this antigen antibody complex, what is done is that to this buffer, washing buffer is added. We all know what is a washing buffer. Washing buffer is basically a buffer which contains a non-azide, non-mercury buffer, which is not going to interfere with the antigen antibody reaction, and it is going to wash away all the unbound antibody from the microtiter well. So those antibody one which are not bound, they will be washed away. Now to this, what is added is another antibody 2 which is conjugated with a enzyme. I forgot to mention the enzymes. Which types of enzymes are used over here? The enzymes which are used in the ELISA test, okay, there are 3 examples which are given in most of the reference books. One is alkaline phosphatase, one is horseradish peroxidase, and the another is beta-galactosidase. Beta-galactosidase and alkaline phosphatase are the enzymes which are derived from the bacterial source, whereas horseradish peroxidase is obtained from the horseradish, that is the eating radish. Okay, so this antibody 2 is linked with or conjugated with an enzyme. This antibody is added to the microtiter well. In a microtiter well, there are 96 wells that are present. 96, around 90 to 96 wells are present. To all those wells, what is coated on those wells are the antigens. to all the wells what are added. Okay, nextly is a sample of that individual who we doubt it might be infected with the HIV virus, for example, and if his sample contains the antibodies, those antibodies we are going to call as the antibody 1. Those antibodies come and bind to the antigen, and the antigen antibody complex is formed. To this we will add the antibody 2, which is conjugated with an enzyme. Now when this is added, after the washing we will add this, and again we are going to wash. Okay, so this antibody 2 comes and binds to the AB1. Okay, because AB1 is bound to the antigen, so AB1 is bound with the antibody 2 and the enzyme is present on the antibody 2. To this complex we are going to add a substrate, okay, against this enzyme. We all know that what is enzyme substrate. So when the substrate is added to this enzyme, the enzyme substrate complex gives a color change. This color change or the absorbance is measured by a gadget that is called a spectrophotometer. Okay, so this spectrophotometer within no time measures all the 96 wells and gives the absorbance of the light. So absorbance of the color. So if the color is present, it means that the whole complex is formed. Okay, and when the whole complex is formed, it means that the antigen antibody reaction has taken place and the patient's serum which we were testing for antibody is positive for the antibody against this antigen. This antigen might be any viral antigen. It need not only be HIV, but it might also be some other viruses against which we are checking for the antibodies. Okay, now this HIV virus which is present, we all know the structure. If you know the structure of HIV virus, on the outer side it contains glycoproteins and oligosaccharides and polysaccharides. So against those glycoproteins and polysaccharides, our body had produced the antibody. I mean, the patient's body had produced the antibodies, and those antibodies have made a complex over here. So this is about the indirect ELISA. Now, suppose if someone is doubted that that person is under a zone of whether he is going to have some infection, okay, within 6 weeks of the infection or after 6 weeks of the infection, the person is tested for the HIV test by the process of ELISA. So this is an enzyme-linked immunosorbent assay. Now the next one is called as a sandwich assay. Now what is a sandwich assay? We will study. Okay, I hope you have understood how the antigens are coated in the indirect ELISA. But in case of sandwich ELISA, what is coated on the microtiter well are the antibodies. What are coated are the antibodies. To this antibody, a sample containing antigens is poured into the microtiter well. So better I will draw it like this. The antibodies are present, and if the patient sample contains antigen, suppose एक patient और उसको HIV infection हुआ है, अगर उसके ब्लड सैंपल में अगर HIV का वायरल पार्टिकल प्रेजेंट है और ये एंटीबॉडीज हमने HIV के अगेंस्ट के यहाँ पे लगाए हैं, तो वो जो एंटीजन है एंटीबॉडी के अगेंस्ट आके बाइंड करेगा. टू दिस अगेन द वॉशिंग बफर इज एडेड. दिस वॉशिंग बफर रिमूव्स ऑफ the remaining antibodies or antigens from the complex or from the wells, and to this what is added is antibody 2, which is conjugated with an enzyme. Now this antibody 2 will not come and bind to the antibody 1, okay, as was in the case of indirect ELISA. This antibody 2 conjugated with the enzyme comes and binds to another okay, to another epitope on the antigen. And so that's why I told you what is an epitope and a paratope. Epitope is a site on the antigen to which the antibody is going to bind. So antibody 1 is bind to another epitope, antibody 2 is bound to another epitope, and to the antibody 2 the enzyme is added, and to this finally the substrate is added, and if the enzyme substrate reaction takes place, the color is going to change. This is in case of the sandwich ELISA. Now it is also called a sandwich because you can see that there are two antibodies and the antigen is in the center. So it is making a sandwich. Okay. Next one is a competitive ELISA. Let's see what happens in the quantitative ELISA. Now, okay, so as I have written, on the microtiter well or inside the microtiter well, antigens are coated as that was in the indirect ELISA. Now to this, what is added is that the patient sample is taken which contains antibodies. This patient sample is mixed with a solution, okay, which contains antigens, and it is incubated for some time. Incubate karna matlab at a proper temperature it has to be maintained. So the antigen antibody complex takes place in the incubation conditions only, and that antigen antibody sample which contains the antigen antibody complex, it is poured into the microtiter well. Once it is poured into the microtiter well, what is done is that the remaining antibodies which have not bound with the antigens, they are allowed to bound with to the antigens that are present in the microtiter well. Okay, am I clear? Antigen antibody complex is made in the solution in the incubated form. That sample with the antigen antibody complex is poured in the microtiter well. Then it is allowed, the remaining antibodies are allowed to bind with the antigens, and then it is washed with the buffer, okay, with the buffer, so that the remaining antibodies are removed off. Okay, so this was about antibody 1. Now what is added over here is antibody 2, which is conjugated with the enzyme. This antibody 2 conjugated with the enzyme comes and bind to the isotype on the antibody 1, and the enzyme is added over here, and then the substrate is added over here, and then the enzyme substrate are allowed to react, and if the color changes, it means that the test is positive. So this was about the competitive ELISA test. The last remaining test is a chemiluminescent test. Okay. Let's see what is the chemiluminescence test. Chemiluminescence means when the enzyme substrate react. Okay, in the previous ELISA test, the color change was being observed, but here light is going to emit. Okay, and that's what is called as a chemiluminescence. The substrate which is used over here is not a chromogenic substrate. Okay, in the ELISA test, the chromogenic substrate was used. In this test, the substrate is called as a luxogenic substrate. That is the light generating substrate. That is a luxogenic substrate. Now what happens here? There is one substance that is called as luminol. Okay, luminol is oxidized. Okay, in the presence of hydrogen, sorry, not water, it is H2O2. In hydrogen peroxide and in the presence of an enzyme that is horseradish peroxidase, this luminol can be oxidized, and when the luminol is oxidized, that reaction is going to emit some light, and that's why the reaction is called as a chemiluminescence. Antibody plus the horseradish peroxidase, which is conjugated with this antibody. Okay, antibody, and there is also antigen. So antibody antigen complex is going to take place. To this antibody, what is conjugated is a horseradish peroxidase, and to this the substrate is added, that is the H2O2, and the luminol is going to oxidize, and that is going to give the it is going to give, or it is going to emit the light. So it is 100-fold more sensitive than the color change test, that is the ELISA test. And if some enhancement agents are added, it becomes 200-fold more sensitive than the ELISA test. So this was about ELISA test, that was indirect ELISA. Then there was sandwich ELISA, competitive ELISA, and the another form, that is the chemiluminescence. I hope you have understood, and I hope that you will give me the subjects and topics so that I can make more good videos for you people. Thank you very much.